Reshma, you can proceed, ma'am.
Yes, sir. Respected pattern, co-pattern, esteemed resource person, respected faculty members, research scholars, participants, and my dear colleagues, good evening to one and all. On behalf of the PG and Research Department of Biochemistry, Sacred Heart College Autonomous, Tirupattur, it's given me an immense pleasure to welcome you all to today's third day section of the 5 days online faculty development program on advanced trends and innovations in modern life sciences, organized as a part of our Platinum Jubilee celebration. Now, I request Dr. V. Gopalakrishnan, Assistant Professor, PG and Research Department of Biochemistry, kindly introduce today's esteemed resource person.
Thank you, Rashmi, for giving this opportunity. Good evening to one and all. I am so blessed to be with you this evening and to have the chance to introduce our today's resource person, Dr. Arivarasu Natarajan Anbazhagan, Research Assistant Professor, University of Illinois, Chicago College of Medicine, Department of Medicine, Division of Gastroenterology and Hepatology, Chicago. Sir did his master's degree and PhD in Aligarh Muslim University, Uttar Pradesh, India. And Sir has served as a postdoctoral research associate in University of South Carolina on osteoarthritis and in University of Illinois, Chicago on epithelial ion and nutrient transport. In 2022, he got implementer of breaking the bias habit by bias reduction in academic medicine program. And the year 2024, he got implementer of bridge program certification at the University of Illinois, Chicago. And Sir acted as a professional member in American Physiological Society, American Gastroenterological Association, Faculty of 1000, and International Society of Computational Biologists and Society of Biological Chemists, India, and many more. And he was honored by various awards like Citizen of the Year, Take the Research Awards, Scientific Excellence Award, Invitrogen Science Hero Award, and many more awards. And Sir published nearly 65 research articles in peer-reviewed journals, and also he wrote more than 20 book chapters, and also he presented his research findings in various national and international conferences. For this presentation, he received several Best Presentation Awards. And he acted as a faculty fellow, research supervisor, lab supervisor for the HS students, master students, PhD students, research associates, postdoctoral fellows, and assistant associate professors. He got US dollar of 73,000 for his research projects as a grants, research grants. He also involved in various administrative services in Illinois University, like a member, host, vice chairperson, coordinator, scientific member, or organizers in various association activities. With this brief introduction, I welcome Dr. Arivarasu to share his valuable insights on post-transcriptional regulation on intestinal ion transporters by microRNAs. Thanks for the opportunity. Now I hand over the session to Dr. Arivarasu Natarajan Anbazhagan. Kindly proceed, sir. Thank you.
Good evening, everyone. And thank you for a very generous introduction. And thanks, Dr. Niaz, for this invitation. And it's my pleasure to present my work today. And as Dr. Govindaraj mentioned, I'm going to talk about the post-transcriptional regulation of intestinal ion transporters by microRNAs. and I have no financial and commercial disclosures. Today, I want to give a brief outline about, like, I want to tell you a little story about my journey and microRNAs, inflammatory bowel diseases, downregulated in adenoma DRA, putative anion transporter PAT1, and fecal microbiota transplant. So my little story begins from Ramakrishna Higher Secondary School in Tirupatur, where I did my 6th to 12th schooling. Then I went to Islamia College, Wanembadi, to do my BSc in biochemistry. Two things which impacted me more in Islamia College. One was Liagat Ali sir, my lecturer, and who used to always mention about the Asia's second biggest university library and the Aligarh Muslim University campus. And the second thing which impacted me was a motivational lecture from an old student named Madesh, Dr. Madesh, who came from USA and shared how he went from Islamia College to USA and got involved in the colon cancer research. So keeping these two things in my mind, first I went to AMU, Aligarh Muslim University, and I did my master's in biochemistry and followed by PhD in biochemistry. And I was interviewed for my postdoctoral position there for US. And in 2008, I went to US and continued doing research. So this is my journey from Tirupati to Chicago. And from 2008, I am here in Chicago. And when I went to US, I was exploring many hot topics to pursue my research. And I was exposed to microRNA research by my mentor. And I felt very excited seeing the amount of research going on in the microRNAs. So if you can see this figure, it shows the number of publications per year when you type the word microRNA in the PubMed. So in this 17 years, I am proud to mention that our microRNA research has led to 3 bigger merit grants. And due to my interest in microRNA research, I was continuously attending more conferences and presenting my research work on microRNAs, and also met a pioneer of microRNA research personnel and became very close to each other. And one fine Monday morning, I woke up and from the bed, I saw scrolling the news. I saw an award announcement and I immediately congratulated him for his award. And he replied back. The person who replied is none other than the person who I know for years and attended conferences together. And he was Dr. Victor Ambros, who won the 2024 Nobel Prize in Physiology or Medicine, sharing with Gary Ruvkon for his phenomenal discovery of microRNAs. And as you all know, a Nobel Prize is considered to be the highest honor in the field of science. If discovery of microRNA gets a Nobel Prize, let us see what are microRNAs. So microRNAs are endogenous, non-coding, single-stranded RNA of 15 to 22 nucleotides encoded by short inverted repeats within the genome. And so far, like 48,000 microRNAs have been cataloged. You won't believe, when we started our research, there were only few hundreds of microRNAs cloned and sequenced. And these microRNAs are known to regulate more than 60% of the genes in the cell. And microRNA gene is found inside the nucleus, which leads to the formation of a primary microRNA. And the enzyme DROSHA converts it into the premature microRNA. And the premature microRNA is then exported to the cytoplasm, where that is converted by dicer to form a mature microRNA. And microRNAs, once the matured microRNA is formed, they can bind to either 5' untranslated region or to the 3' untranslated region. And when they bind to the 3' UTR with perfect complementarity, they lead to mRNA degradation. And when their binding is imperfect, they lead to translational repression. So with this mode of idea, we came with a working model where we will transfect a mimic in the intestinal epithelial cell line. This will indirectly increase the microRNA inside the cell, and the mature microRNA will then bind to the 3' UTR of the target mRNA and decrease the protein expression, what is the target of. First breakthrough in microRNA research came from University of Chicago by Dr. John Kwan, who came from Harvard to Chicago, where he showed a differential pattern of microRNAs in the human biopsy from active and inactive IBD patients. So, what is IBD? So, IBD constitutes of Crohn's disease, which is a condition of chronic inflammation, which potentially involves the all location of gastrointestinal tract, but it frequently affects the small bowel and the beginning of the large bowel. So the normal symptoms include the loose or watery stools, cramping and abdominal pain. And when it comes to ulcerative colitis, it is a chronic gastrointestinal disorder that is limited to the large bowel, like the large intestine more than the small intestine. And patients with ulcerative colitis are at higher risk of colon cancer. And to date, if you see 3.1 million persons are affected in US and 2.2 million in Europe, and it was more alarming to see in India, it will be more than 2 million IBD cases by the year 2035, and which is a fold 4 increase from today's data point. And again, as I mentioned, the main symptom is diarrhea. And when it comes to diarrhea, we can immediately pinpoint that the diarrhea is formed due to the disturbances in the electrolyte balance of intestinal ion transporters. As we all know, the intestinal ion transporters play crucial roles in cellular function, such as metabolic process, osmoregulation, water and electrolyte homeostasis, and nutrient absorption and neurotransmission. And we were more interested in starting with the two important chloride SLC26 gene family members, which are involved in the major luminal chloride bicarbonate exchange. One is putative anion transporter 1, another one is the downregulated in adenoma, which is also called as SLC26A3. So the downregulated in adenoma is considered to be a novel susceptibility gene for IBD. The studies from the Japanese population, as well as the Chinese population, as well as of the Icelandic population, all showed that these IBD patients showed much decrease in the DRA, and they considered it as a novel susceptibility gene for IBD using this genome-wide associated studies. And there are more proof that there is a reduction in the expression of DRA in the colonic crypt of chloride bicarbonate exchange activity with a parallel decrease in the mRNA, which has been reported in both ulcerative colitis patients in IBD. So the downregulation of DRA function and expression has emerged as one of the most critical events in IBD associated diarrhea. Not only that, the mutations in DRA caused a rare genetic disorder called congenital chloride diarrhea, which was exhibited by a voluminous diarrhea and with a dilated bowel, as you see, with a distended abdomen, which is also a cause of metabolic alkalosis, dehydration, and defect in chloride absorption. And you can see the patients normally exhibit a fecal chloride concentration of more than 90 millimolar per liter, where the normal is only 10 to 15 millimolar in the normal conditions. And these studies highlights the importance of DRA gene. And so far, the DRA regulation has been shown at post-translational level using the signaling, trafficking, and lipid rafts, and by transcriptional regulation using the transcription factors. Whether the modulation of DRA occurs at the post-transcription level was not known. And we were the first group to investigate the effects of the putative microRNAs on DRA expression and its 3' UTR. So we followed the methodology where we cloned the 3' UTR of DRA in a very special Mir-Glow vector, which added dual luciferase, and we implemented the in silico analysis, and we utilized the KCO2 cells for the present study where the DRA mRNA expression was determined by qualitative PCR and Western blotting were done for estimating the protein and the luciferase for the transfection. In the first set of results, we saw a significant decrease in the relative luciferase activity, indicating that some factors are binding to the 3' UTR of DRA. It could be microRNAs or it could be RNA binding proteins. But we first wanted to explore the microRNA first. Using the in silico approach, we utilized 5 different algorithms to identify the microRNAs that can potentially target DRA. As you can see, MirDB, Miranda, Mirgen, microcosm, and target scan. Since target scan showed the highest number of 65 targets, we used target scan algorithm, which showed highest number of candidates, and you can see throughout the 3' UTR, we can see several microRNAs binding and the respective area on the 3' UTR. Based on the target scan, the context score, we selected the top 5 microRNAs which showed the highest context score. More the negative free energy means more potential to bind. And these 5 microRNAs were considered to be the top 5 candidates with higher potential energy to target DRA. So in the first set of experiment, when we co-transfected the 3' UTR with the respective mimic, unfortunately, none of the microRNA mimic, these 4 of them, didn't show any change in the relative luciferase activity. It means these microRNA mimics or the microRNA is not binding to the 3' UTR of DRA. But to our surprise, when we co-transfected the 3' UTR of DRA with the mimic 494, we saw a significant decrease in the luciferase activity, which showed that microRNA 494 is binding to the 3' UTR of DRA. With that, we wanted to also authenticate the proof that when you are transfecting the microRNA levels are going up. So we checked the levels of microRNA 494 when we transfected with microRNA mimic. So as you can see, there was a significant, more than 3-fold increase in microRNA mimic of the 494 transfected cells. We checked the binding region of microRNA 494 in different species. For example, these are the 3 models what normally we use to compare with. So we checked whether the binding region of microRNA 494 is conserved on the different species. So as you see, the binding region of microRNA 494 is conserved in rat, human, and mouse. If the binding region is conserved, it means the microRNA is more functional. Next, we checked the mRNA levels of DRA. And it was like a surprise for us, and we were very upset that there were no change in the DRA mRNA levels. This upset is because the central dogma always explains that any increase or decrease should be started with the mRNA levels. Since there was no change in the mRNA levels, we were a little upset, but we wanted to proceed to the protein level. To our surprise, we saw a significant decrease in the DRA protein expression. And again, we felt that we are stuck in this part because the protein is decreasing, but there is no change in the mRNA. And this particular project went for halt. We didn't do any research for a couple of months, and we were just thinking about why this has happened, how we can fix this. And then, luckily, we just come to know Dr. John Kwan, whom we collaborated. If you remember in the first slide, I showed that he was the pioneer who first mentioned the differential microRNA pattern in IBD patients. So that was a very phenomenal breakthrough in the microRNA research. And luckily he was in Chicago. So I started collaborating with him, and he mentioned that he had some similar type of results. And then he asked to proceed, and then with this collaboration, we proceeded next to the next level. And as you see, there is a significant change in the protein. In this 15 years, we became very close friends. And then when we last met, you can see the significant change not only in the protein, in us also. So when he mentioned that you can proceed with the research, that was a big boost, and we continued the research where we wanted to check the effects, whatever we have seen in KK2 cells are cell line specific, or these effects are also seen in other cell lines. When we checked these effects, we transfected the 3' UTR of DRA in other cell line, intestinal epithelial cell line called HT29, where you can see a similar result of significant decrease in the relative luciferase activity. And when we co-transfected with the mimic, again the mimic showed a significant decrease in the relative luciferase activity, which very clearly indicated that the effects, whatever you have seen in KK2 cells, were not cell line specific. Then we next moved to prove the concept that if everything is happening because of microRNA 494, what happens if we mutate the sequence in the 3' UTR? Using the site-directed mutagenesis, we completely mutated the 3' UTR of DRA where the microRNA 494 binds. As you can see, the first two bars in all three cell lines showed that there was a significant decrease in the co-transfection of 3' UTR to mimic 494. But this effect was completely blocked when we used the mutated 3' UTR instead of the normal UTR. So to summarize, we thought that according to our results, when we transfected the mimic 494, it increased the microRNA 494, which led to the binding in the 3' UTR of DRA, and which inhibited or decreased the protein expression of protein. And after that, this final experiment, we thought that we are done and ready for publishing. And since nobody has ever told this mechanism of post-transcriptional regulation of DRA, we were very excited to publish. Then we came across a publication where they did some of the microRNA experiments. They treated the cells with ATRA, and this ATRA increased microRNA 494 levels starting in a time-dependent manner for 24 hours, 48 hours, and 72 hours. Since we were trying to tell a very important mechanism to the world, we felt that further authenticating our result as a positive control, we wanted to treat our cells with ATRA and then see what happens to the microRNA 494 levels and the DRA levels. But to our surprise, we got a negative result. What we got is, we treated ATRA for 8, 16, and 24 hours, and there was no change in the levels of 494 microRNA, but we saw a significant increase in the mRNA levels of DRA. And as I earlier mentioned, DRA is a very important chloride bicarbonate exchanger. Any tool or any agent which can increase DRA expression, then it is a therapy. So we were very excited and we wanted to explore what is ATRA. It is simply a vitamin A derivative. It is a natural derivative, and it is a potential regulator of growth, development, and differentiation. And it also downregulates the inflammatory responses in human ulcerative and the murine colitis. With this, we did enormous amount of research in this area where we showed ATRA using the transcription factors HNF4, HNF1 beta It binds to the it binds to the DRA promoter region and increases the DRA mRNA levels and finally increasing the DRA. We wanted to put a question, since the ATRA is an anti-inflammatory, whether the DRA expression can be abrogated or ameliorated by ATRA in the inflammatory models. For this, we used the black 6 mice where we exposed them to 3% of dextran sodium sulfate, which is a known agent. When given in drinking water for 7 days, it developed an acute colitis-like model, which is a very well-approved model. And the ATRA 1 milligram per kg body weight was given, and we did some in vivo imaging, and the mice was sacrificed on the 8th day, and we scrapped the colonic mucosa and looked for the pro-inflammatory cytokine profile, DRA, mRNA, and protein levels, and immunostaining. As expected, whenever you feed the DSS, the dextran sodium sulfate, in drinking water, the animal tend to lose body weight. And as you see, when the control animal, the green line shows there is no change in body weight. But if you see the red, which shows a significant decrease in body weight, but when we have given ATRA to the DSS, you can see there is no change in the body weight further, but it showed a little bit of increase. It shows that ATRA avoided the sickness of the animals. And we also wanted to see what happens to the inflammatory levels of myeloperoxidase activity, which is an indicator of inflammation. As you can see, there is no any myeloperoxidase activity in control and ATRA, and the DSS significantly increased the myeloperoxidase activity, showing that there is more inflammation, and this inflammation was significantly ameliorated by ATRA treatment. And for the first time, we also tried to show the in vivo imaging about the live animals where we can trace the inflammation using a probe L012. We tried to show that the DSS animals showed a significant the inflammation according to the luminescence, and the ATRA, the last mice, showed significantly decreased luminescence compared to the DSS group. And we also showed that the protein expression levels of DRA was significantly ameliorated by ATRA treatment. As you can see, the third panel of DSS, where the DRA expression was significantly decreased, which was ameliorated by ATRA, which increased the DRA expression. To summarize this, from the literature and our research experience, we know that DSS-induced colitis leads to the increased pro-inflammatory cytokines, and we have done several experiments to show that it increased STAT3 phosphorylation, which decreased DRA mRNA levels, and which finally decreased the DRA protein levels. But as we treated the animals and the in vitro cell culture, we came to know that ATRA is able to block this process and ameliorated the conditions caused by inflammation. So, as this ATRA project finished, which started from a negative result, that ATRA did not change the microRNA 494 levels, and reversely it increased the DRA expression. But we were very fortunate that it led to two bigger publications, one in JBC and one in Inflammatory Bowel Disease Journal, and one very big grant. So my simple advice is, please never stop the project because of a negative result. And next, we shifted our gears to the knockout models. So the DRA knockout mice, as you can see, the left one is the wild type mice, where you can see the normal cecum. But the DRA knockout mice, which is a global knockout, you can see there is a bulged cecum, which is a very clear indication that this DRA knockout mice, from the beginning, they showed a diarrheal phenotype. And we also wanted to see whether there is any tight junction permeability compromise. So we did a functional test of FTC dextran. The way how we do this experiment is the FTC dextran is a fluorescent dye which is inert. We gavage the animals in the morning of the day of sacrifice, and then after 4 hours we sacrifice the animals and then measure the fluorescence in the serum. As you can see, the knockout animal serum showed a significant increase in the FTC dextran, which is a very clear indication that there is a gut permeability compromise in these animals. We also did untargeted metabolomics from the feces, colonic mucosa, and the serum, which all showed a similar pathways which were affected in the DRA knockout mice, which were involved in the short chain fatty acid metabolism, bile acid metabolism, sialic acid metabolism, and aromatic amino acid metabolism. And after this metabolomics, we also wanted to see what happens in this mice in the immune profile. As you can see the nanostring panel of immune profile, the DRA knockout mice showed a increased expression of many in immune cell signature, which tended to inflammatory signature, like increase in the leukocytes, natural killer cells, and macrophages. And we also utilized the top cutting edge model of organoids, where we showed the organoids from the wild type mice and the organoids from the DRA knockout mice. It showed a very increased expression of IL-33, which further proved that there was an inflammatory immune signature which is elevated in DRA knockout mice. And we also showed that the DRA knockout mice not only changed the metabolic profile and the immune profile, there was a compromise of tight junction permeability. We wanted to see what happens to the occludin, a tight junction protein. As you can see, the green color in the wild type, there was occludin, but the DRA knockout mice, it was significantly lost. This very clearly indicated that the compromised barrier integrity is due to decrease in the occludin and the key tight junction protein. And this research of ours ended in a gastroenterology paper in 2021. And as you can see this model, there is a gut microbial dysbiosis, and there is a compromised barrier integrity, there is a thin mucus layer. We know that so many things are happening in this model, but we were not very sure about the mechanism. So we went for microRNA panel, like on transcriptomics. We went for microRNA sequencing here in the colonic mucosa of DRA knockout mice. As you can see, the downregulated microRNA, there were 3 microRNAs which we found downregulated, and there were many microRNAs which are upregulated. And we are currently proceeding with this microRNA profile and their targeted microRNA genes, which can effectively be the mechanistic overrule for the observations what we have made in the DRA knockout mice. So this finishes the DRA knockout portion. And now we are moving to the putative anion transporter, the PAT1, which is also called as SLC26A6. So the putative anion transporter 1, it's a chloride bicarbonate exchanger again, but it additionally can secrete oxalate and sulfate, and it plays an important role in transcellular oxalate transport. And this knockout mice in the research earlier, people have shown that the global knockout of PAT1 exhibit hyperoxalemia, hyperoxaluria, and high frequency of kidney stones, which poses a major risk for nephrolithiasis, which is also called as kidney stone disease. For this, we first cloned the 3' UTR of the PAT1, and we performed a similar set of experiments like that of DRA in KCO2 cells, HT29, SKC015, and T84, and we found that in all the cell lines, the relative luciferase activity was significantly decreased in 3' UTR, and the target scan showed the key microRNAs which can bind to the 3' UTR of PAT1. And we selected the top microRNA, the 6 of them, and then we transfected with the 3' UTR of PAT1. And as you can see, two microRNAs, the 125-5AP and 423, showed a significant decrease in the relative luciferase activity, which made us to select these two microRNAs and proceed. And the 423 didn't change the protein levels. So we tried to proceed with microRNA 125A5P. We found, if you remember the DRA data, we were stuck with the project and we were very upset that the mRNA levels of DRA didn't change. But to our surprise, the PAT1 mRNA levels changed. So we had two projects where microRNA mode of action, two main mechanisms. One, mRNA degradation, another one is translational repression. In DRA, we saw translational repression, and here in PAT1, we saw mRNA degradation, which means the 3' UTR of PAT1 and the microRNA 125AP, their binding is perfectly complementary. And we finally saw the decreased protein expression of PAT1, and we felt very happy that this is a different mechanism than what we have found earlier. And this is a very important proof that the post-transcriptional regulation of PAT1, which we were again the first group to mention this regulation, and we mutated the 3' UTR PAT1 where the microRNA 125A5P can bind, which significantly decreased in the normal UTR, and this was blocked when we mutated the site. And in this particular model, when we transfected the cells with mimic 125AP, which increased the 125A5P, and which bound to the 3' UTR of PAT1, leading to the decrease of the PAT1 protein expression. And the microRNA, now knowing that microRNA 494 and the microRNA 125AP are two very important targets which negatively regulate DRA, we came with a therapy for using it as a target therapy where we can use the 3' UTR and the negative inhibitors or the inhibitors of both MIR 594 and MIR 125-5P, which will block the binding of the microRNA to the 3' UTR and will increase the DRA, and which will finally increase the chloride absorption. And so far, the regulation, whatever we saw with the DRA and PAT1, the PAT1 and IBD story was very novel because only our group has earlier shown that the reduction in PAT1 expression using interferon gamma earlier. But recently, there was a study which showed that PAT1 was significantly decreased in the IBD patients with the Crohn's disease as well as with the ulcerative colitis. That was a very big encouragement for us because we were able to now connect IBD and nephrolithiasis, where we are targeting a single protein for handling two diseases. And when we checked the knockout PAT1 animals, to our surprise, all the key tight junction proteins, including occludin, Z01, and beta-catenin, they all showed a significant decrease in the protein level, which shows this PAT1 knockout animals exhibited tight junction permeability compromise. To prove it functionally, we again gave the animals to the FITC dextran, 600 milligram per kg body weight in the morning, and then we sacrifice these animals. We can see a significant increase in the FITC dextran, mentioning there is a increased leakage that is called as the gut permeability compromise. And this very clearly indicates the proof that there is a compromised barrier integrity in this knockout animals. And when we exposed these animals to DSS, you can see the PAT1 knockout animals exposed to DSS were very vulnerable, and these were the worst group which got very sick compared to the normal DSS. It mentioned that when you knock down PAT1 inside the animal globally, that much worsens the condition of inflammation. As you can see, the first picture shows the normal wild type animals in colon. The second shows the DSS treated mice colon. The DSS mice colon, you can see there is a significant decrease in the colon length. So a decrease in colon length itself is an indication that the animal are sick. And you can see there is no stool in the colon. And when you come to the PAT1 knockout, it, it was much similar to that of the control wild type animals. As you can see, the last PAT1 knockout plus DSS group, you can see the length of the colon was very much shrunken, and you can see even the cecum was very small. Which two are the conditions mentioning among all the groups? This particular group was exhibiting the most inflammatory conditions as per the observations. We next wanted to see what has happened to the HNE staining and the architecture of the intestinal epithelium in the colon. As you can see, the PAT1 knockout DSS was the worst group, which very much distortion of architecture of intestinal epithelium, where the wild type and the PAT1 knockout animals showed a very clear and a well-formed architecture of intestinal epithelium, but the PAT1 knockout DSS group was much worse than the wild type group itself, where you can see an increased crypt damage, where you can see an increased edema and increased inflammatory infiltrate. And one another indication what we wanted to see was the goblet cells. The goblet cells are known to be beneficial. More the goblet cells are better to ameliorate the inflammatory conditions. As you can see, the PAT1 knockout plus DSS group merely had any goblet cells, which clearly mentioned that the cells which are supposed to act as a beneficial and the health maintenance cells, they were much less in the PAT1 knockout and DSS, which clearly indicates that this animal which is exposed to DSS, the PAT1 knockout animals, they are in a very worsened inflammatory conditions. We also saw that the tight junction protein occludin was present in wild type animals. It was decreased in the PAT1 knockout animals, but it was most decreased in the PAT1 knockout plus DSS group, which clearly indicated that these animals have the highest compromised tight junction permeability. We then moved our gears to microbiome analysis, where we found that the PAT1 knockout animals showed a gut dysbiosis, where the principal coordinate axis showed there was a very clear demarcation between wild type and the PAT1 knockout animals, where you can see a significant decrease in the firmicutes and the significant decrease in bacteroidetes, which tells us that in the wild type animals there is increased lactobacillus. They are all called as probiotics. They are beneficial for the health. The wild type animals are having more of the prebiotic type of signature, whereas the PAT1 knockout animal microbiome, they were having more pathogenic profile or the inflammatory profile, where you can see more increased clostridium and the streptococcus, etc. So why we have to bother about the microbiome? As you can see this figure very clearly explains that compared to the human, the microorganisms are over dominating. Yes, nowadays we are calling the microbiome as an hidden organ because the microbiome, they contribute over 150 times more genetic information than that of the entire genome. Sorry, I saw an hand raised. Do you want to ask any question?
Revati madam, you can raise your question at the end of the session, please. Sir, kindly proceed, sir.
Okay. So the microbiome is not only acting more bigger genome than of the human itself, but they were nowadays connected with every single part of your day-to-day life. That clearly indicates that gut-brain axis, whatever you eat, whatever how healthy your intestine is, that is how your brain activities are connected. So every single disease nowadays is connected with gut. So only it is not only the gut brain axis. Nowadays you can see the researchers more extravagantly performing their research in the field of gut renal axis, gut liver axis. So gut is now the main connecting point for all the research, and the reason behind is the microbiome of the intestine. When we saw the microbiome is playing such an important role, we now came back to our previous experiments with our PAT1 knockout animals. What we decided, we will make the wild type animal and the PAT1 knockout animal a co-housing experiment, because if you want to transfer their microbiome, when the two animals are kept in the same cage, they are caprophagic because they tend to eat their feces. So we wanted a very optimal microbiome shift to take place. It takes place nearly in 4 weeks. So we kept the wild type and the PAT1 knockout animals together for 4 weeks. As you can see this result, the pre-PAT1 knockout animal and the post-PAT1 knockout animal, you can see the wild type animal after co-housing has started showing the features which were like that of PAT1 knockout microbiome profile. That was really very exciting for us, because the co-housed wild type animals started showing the tight junction permeability compromise, which were very normal before co-housing. And then we next moved on to the metabolomics. When we did the metabolomic studies in the PAT1 knockout, we felt there was a very clear-cut difference between the metabolites of wild type and the PAT1 knockout in the coordinate axis. And we can see there was a very big decrease in the very important metabolites like butyrate, pyridoxine, and the schemate. And we also saw that there were much increase in the galactose metabolism, and there is a much decrease in butyrate metabolism. So this very clearly indicates there was a very divergent difference in the metabolites of wild type as well as the PAT1. And next, we wanted to see the transcriptomics of this profile. So we did an RNA sequencing between the wild type and the knockout animals, where you can see there is a very distinct difference between the wild type animal and the PAT1 knockout animal RNA, the transcriptomic results, which very clearly indicated that the wild type animals, compared to the wild type animals, there was a significant increase in immune system process, cellular response to the unfold protein, and the protein refolding in the PAT1 knockout animals. Which all this huge data amount of multi-omics and all data, we ended up the publication, which gave us a very hard time of 3 years of work in final publication in the gastroenterology, which at the time had near about 33.3 as an impact factor. And we had a question, why this is happening in the PAT1 knockout. Some reason is microbiome, but we are always thinking to connect the story with microRNAs. MicroRNAs can also play an important role for the observations what we are seeing in the PAT1 knockout model. So we went for the microRNA sequencing using the nanostring panel, where we found the PAT1 knockout colonic mucosal showed a very significant decrease of 5 microRNAs, and several microRNAs were upregulated. To our surprise, when we did an in silico analysis, the upregulated microRNAs, most of them were targeting the tight junction proteins. So this gives us a new hope, and the future studies we are planning to do more studies connecting the story of how the microRNAs are involved in decreasing the protein expression of tight junction proteins in these knockout models. It is not very simple when we talk about microRNA and microbiome, but there is a very clear proof to connect the story, where the literature and the research evidence indicates that In a normal mice, you see a signature of microbiome where you can see the probiotic-like signature is increased, and you can see certain microRNAs are increased. But when it comes to a diseased model of colitis, you see there is an increase of pathogenic-like signature and increase of different set of microRNAs. So this altered microRNAs and the modified microbiome profiles are very clearly indicating that there is a microRNA and microbiome cross-check, and many publications are coming favoring our concept what we are trying. And one important model what we wanted to establish and prove our concept with microbiome and microRNA is creating, either doing our experiments in the germ-free environment or creating a fecal microbiome transplant model. So the main ideology of creating this model is we will try to implant the microbiome of these knockout models after dividing the microbiome of the wild type animals, and the gavaging of this microbiome of the knockout will give you the pattern of the microbiome of the knockout animals. And this is the very important concept, and where we also created an humanized mice model using this concept. And with this final model, I want to conclude, and I really acknowledge my mentor, Pradeep Dudeja, and my team, and my collaborator, Dr. John Kwan and Dr. Ziaudi Tan, and the funding for NIH for the RON and the VA merit grants. And again, a thanks for the Sacred Heart College and Dr. Niaz and the department for providing me the opportunity to present my work today. With that, I finish my presentation. I am happy to take any questions.
Thank you, sir, for sharing your expertise and valuable insights throughout the section. So we now open the floor for the query section. Participants are requested to post their questions related to the section in the chat box.
So, yeah, I can see the question from Dr. Niaz, the role of microRNA in gene silencing. Yeah, so as you can see, the microRNA has not only can do a negative regulation of the gene, it can also do a positive regulation. Yeah, the positive regulation comes when the microRNA binds to the 5' UTR region, but all the silencing comes only when the microRNA binds to the 3' UTR. So when a microRNA binds to the 3' UTR, it simply acts like an saRNA for that particular gene. So the gene silencing story is always negative correlation with the gene, target gene, when the microRNA binds to the 3' UTR. Depending upon where the microRNA is going to bind, the silencing or upregulation of that gene takes place. I hope I answered the question.
Sir, I am Govindarajan. Just a curiosity. So, what is the control drug you used, sir, during the animal studies?
So the control for the
drug, I mean, like that ATRA and this like ulcerative colitis, you use, no? That with
yeah. So the control animals, the DSS is going in drinking water. So the control animals are just having only the drinking water.
No, the drug which elevate the symptoms, like the synthetic drug, like you compare.
Oh, the ATRA? Yeah, the ATRA was given in corn oil. So the ATRA dissolved in corn oil was very effective because we tried several diluents as the vehicle. Corn oil was the vehicle which we were very effectively able to utilize it. So we gave all our animals the corn oil.
Okay, so there will be no any comparison with any synthetic drug which elevate the symptoms comparatively to ATRA, so that you can compare with the significance compared to ATRA. This drug will maybe have more potential or something like that, or compared to the synthetic drug, our ATRA will be have more potential. Yeah, you didn't compare any synthetic drug.
Yeah, thank you for the question. And as I just, this is only a part of my area of interest where I presented this work. If you can Google one of my papers where we have tried to alleviate the TSS colitis gut inflammation using the nanomedicine GLP-1. So
Okay.
this GLP-1 nanomedicine was one of our up to the mark, very fruitful study where the GLP peptide was
Yeah,
very
yeah.
short-lived and known to have a very potent role in alleviating the lung inflammation.
Diabetes.
Yeah, but our group was the first group to mention that the nanomedicine GLP-1 can ameliorate the gut inflammation. So in that,
Okay. Yeah,
this nanomedicine was made using the sterically stabilized micelles methodology, SSM. So when we used this nanomedicine, we were able to improve much of that inflammation created by the TSS colitis, including the expression of DRA, what we have seen. And this is all in the publication where we have committed, where we used the control as the sterically stabilized micelles to the control animals.
So one more question, like any nutraceuticals, maybe like herbal drugs or something which maybe has the potential, like we see as my work in aloe vera. So aloe vera has the more potential in as the elevation of these ulcerative colitis also. So
Mhm.
is there any, because it has more antioxidant and anti-inflammatory capacity. So
Mhm.
likewise, is there any like nutraceutical you may, I mean, you compare, I mean, you just gone through regarding this, or just an, I mean, only with the ATRA, right?
Yeah, that's a nice question. Thank you for the question. Like the every day end of a day in a researcher's life is if some drug which can have very less side effects can fix your disease.
Yeah.
This is the main goal of every single researcher who is doing this.
Yeah.
And my first preference for any commercially available drug, if you compare the commercially available drug to treat a disease with the nutraceuticals, my option will be 100% towards the selection of the nutraceuticals. And first we started most of our studies using the probiotics. So
Yeah.
we have some of our publications where we have shown, particularly the lactobacillus species acidophilus, compared to all other lactobacillus species, is very effective against the DSS gut inflammation. And we have also shown the lysophosphatidic acid, a bioactive lipid, and the sphingosine 1 phosphate, a bioactive lipid. Again, they are more effective against in the treatment of ameliorating the gut inflammation. So
Okay.
first we started with the probiotics, then
Yes.
we did the ATRA. The ATRA was a mere accident because
Okay.
we were not thinking of we didn't do the experiment of ATRA just because of it increased the DRA. We wanted to show that the microRNA 494 is going up, but we never even imagined that that will end up in two publications. That was like a 4 years of project ending up in a grant.
Okay.
Yeah.
Thank you,
So
sir.
first,
Thank you.
thank you.
Thanks. Thank you. So, any questions from participant sides?
Thank you. Some of them has raised hands. If they want, you can directly interact with our speaker. So Sar is pointed out a word during his response to Govind sir, researcher, on behalf of the audience. I think see some of them. I think Imran Ahmad Malik from Kashmir. I think so. Regarding the researcher, they want to switch over their USA. There is the entry in the USA easy for a researcher, sir, nowadays. What about the TOEFL and other? It is not a technical question. I'm not because I'm not well versed like Govind sir, who interacted with you subjectively. My question is, it is general, in general, about the entry into USA. What about the requirements? What about the TOEFL, etc. for the researcher to move? Because some researchers I seen, some of them
Mhm.
doing PhD and postdoc. Please, sir.
Yeah. Yeah, so that is like again a futuristic question, and it is the question I hope I had when I was doing my bachelor's degree. Because whenever you ask a person what you want to do after BSc biochemistry, I don't know how immature I was at the time to tell that after finishing BSc Biochemistry, I told that I will go to US. But I don't know how to go to US and what to do for that. And this was a bigger question. But whomever asked where you want to go, I want to just tell that, yeah, after finishing biochemistry, I will go to US. And the question compared to before January and after January, there are two different phases in the research entering US, because now they have changed the visa related process for the H1Bs. But for the student visas, there were no any restrictions. As for today, the students who want to study outside, the students who want to pursue their either the masters or the PhD, still the same route what has been followed from the day one earlier days is the same. There is no any change in it. So if you want to come to US for doing a postdoc, that is completely different. You don't have to do a TOEFL or anything. You can, after your PhD, all what you have to apply for the positions where they are, and if any of the scientists interested in your CV and your work, they can provide you an opportunity. Earlier, before 20 years, this was the task. You have to email a random scientist who is interested in your work, and they will call you. They will reply. If you get a reply, if I emailed 600 scientists, I got reply from few of them. So that is the normal, because I was not able to understand, and I was always getting angry why this scientist is not even replying my email. I took a time to prepare this area of interest, and I have emailed him 3-4 paragraphs. They are not even replying. I felt that that assumption what I was thinking was very wrong, because at very earlier stages, even in my because I was an instructor for near about 6 years. When I was instructor in the same department in University of Illinois Chicago, I used to get like 50 emails from the postdoctoral fellow looking for the postdoctoral position all over the world. And I was just thinking, as an instructor, when I can get 50 applications, I was thinking about the other people, like the professors who are having the grant, how many applications they will be getting. But I always want you to encourage, until or unless you are not going to stand in the line, you are not going to get your number. So you have to try. And before getting into US, I got 2 postdoctoral positions, one from Japanese Health Science Foundation in Japan and one from Karolinska Institute in so Karolinska Institute was like a very important one, but both of them I didn't have my PhD degree at the time, so I was supposed to turn them down, and then I then came to US after finishing it. So you have to take, if you want to do masters, then your requirement to enter US are different. Please, nowadays the tools are very easy available on any Google and any chat GPT. You can mention what are the steps you have to follow to enter into US, to perform your or to pursue with your master's degree. It will do you get stepwise information guidance. And most probably, I don't think so whether that opportunities are there in Tirupatur or any nearby towns, but 100% sure I know people who have been continuously coming from Chennai and all other places, all the metropolitan cities. It means they know the line what has to be done. There are some centers where they guide you how to prepare for this, because the applications starts way earlier. Because coming from a small town, I had the same inhibitions, where to ask, who to ask, what to apply. So these things. But one very important thing what I want to tell to the all audience and the students who are here is, in my time point, there was a fashion of coming to US to do master's degree. But now the science and technology, and due to the development of this internet and everything, I have started seeing the students from India coming to do their bachelor's degree here. And I talked with one of this bachelor's degree student who came from India to the Michigan University, and then I asked when you started the application, because after 12th you came here for doing your bachelor's degree. He told that after finishing the 10th, in the 11th beginning, my parents started putting the application. So I was just thinking, when even doing my MSc, I was not very clear where to apply and what to do. But nowadays, if you are not aware about the tools which is available, because nowadays the knowledge is no more like a very unlimited source, only very few people can get. Every single person sitting in any corner of any world can get the same information what a person in US can get. So I will encourage every single person to pursue, to get or increase the awareness about the programs available outside. When you start searching, then only you will end up with 4 wrong things, then you will finally end up in one right thing. But everything needs a commitment. So I will encourage everybody. And I tell, this is, US is an opportunity of land. So where always the opportunity is, whomever comes with the potential skills are always appreciated. I want every single person who wants to come and learn things and do much in research, pursue their career in this line. I always encourage them just to know additional things what they want to know about it. And I can be of any help. My email ID is with Dr. Nias, and you can always share that. And I will be happy to help if I can do anything from my side.
Sure, sir. Thanks a lot, sir. Over to Reshma or Govindrat, who is handling it? I don't know.
Once again, we thank you, sir, to accepting our invitation and share your expertise and insight throughout the section. Then, thank you all for your presence and cooperation. Let us look forward to an enlightening and enriching section for tomorrow. Thank you all.
Thank you, sir. Thanks a lot. Rumba thanks. From my
Oh,
bottom
Thank you.
of the heart. Thanks a lot.
Thank
Thank
you.
you. Thank you.
Thank
Thank
you
you.
for
Yes, sir.
Thank you. Thank you, Reshma. Thank you, Govind sir. Sheila, thank
Thank
you.
you.
Thank you, sir.
Bhargavi, thank you.
Thank you, sir.
Good night. Good night. Good night.
Good night, sir. Thank you.
Avarku good morning.
Or good morning.
Sir, good morning, sir. Okay, sir.
Thank you, all of you. Thank you. Thank you.
Okay, thank you.
Okay, thank you, sir. Thank you, sir.
Thanks.